Welcome back to this course on organic chemistry in biology and drug development. The first half of this lecture series, we have discussed the different biochemical processes where organic chemistry plays a dominant role, and once we have finished studying those different life processes, different processes in the microorganism and also in human body, then we started the next part of the course, that is organic chemistry in drug development. So, basically this needs a very solid foundation of biochemistry knowledge, that means organic chemistry in the biological system. Now, in this drug development, today is we are almost finishing this second part also. Few things are remaining, we are going to cover that, but before that I want to just summarize what has been done till now in the area of drug development. So, initially we discussed that strategies to develop, to design a drug, okay. And we have seen the importance of target identification, validation, computational chemistry, then which is also called in silico screening, followed by identification of a hit, then the lead, then lead optimization, preclinical studies, and finally, the clinical studies involving human beings, human subjects. Okay. And if everything goes okay, then the drug is approved by the Food and Drug Administration Authority of it is that authority in USA is considered as the best screening method, because they approve the drugs only on the basis of all these parameters that we have discussed. Okay. At every checkpoint they will see the data. Okay. Now, then we went for that how to quickly achieve a heat. So, you have to have a large library of compounds, and we discussed the combinatorial chemistry at that time, okay, large library of compounds. We have also discussed what is called the safety margin of drugs, safety margin, okay, safety margin of drugs. That means, every drug has a side effect and also is toxic after certain amount of the dose. So, we have to be very careful about the therapeutic index of a drug, because that is the margin which is allowed or which is allowable to take. Okay. So, that we have discussed the therapeutic index as a ratio of ED50 in terms of ED50 in terms of your LD50 divided by ED50, that means effective dose for 50% of the population, lethal dose for 50% of the population, or it could be TD50 by ED50. That is the therapeutic index. The higher the therapeutic index, the safer is the drug. Okay. The D is, then we went for the different classes of drugs. Okay. So, one is first the neurotransmitter we discussed. Then we have discussed, so at that time we discussed acetylcholine, ACHE, acetylcholine. We have also discussed the dopamine. So, when we have discussed all these, we discussed their production and their degradation and whatever effect they produce when binding to the receptor. Then we discussed we have taken the antimicrobial compounds. Maybe before that, I think before that we have neurotransmitter, and then followed by we have taken the antihypertensive drugs. I think that is in between these two, antihypertensive. And why we have picked up antihypertensive drugs here? Because the example that we have studied is what was called the angiotensin converting enzyme and and a hormone which is called angiotensin 2. So, that is why we have put. So, this is the hormone related hypertension and this is neurotransmission related brain dysfunctions. Okay. Then we discussed antimicrobial compounds in detail. We have discussed the penicillin, the sulfonamides, the cephalosporins, the tetracyclines, the other erythromycin, everything was discussed here. Okay. Then we discussed the polyketides in connection with the erythromycin, polyketide biosynthesis. Okay. Because a majority of the drugs that we use are natural products and a majority of them are polyketide origin. After polyketide, we discussed the antiviral drugs, and once we are through with the antiviral drug strategy, design strategy, then we went on to discuss anti-cancer agents, HI, and the last session we discussed the anti-ulcer. Okay. Anti-ulcer agents, which are basically nothing but H2 antagonist, that is one aspect. Another is proton pump inhibitor. Okay. In the last session, we have discussed the chemistry of ranitidine and also the chemistry of omeprazole, proton pump inhibitor. So, we will now move on to another aspect which is also related to hypertension, which is also related to hypertension, because hypertension is the basically the pressure, the blood pressure is higher than the normal level, that is what is hypertension. Now, hypertension can be due to several factors. One of them I said the receptor problem, angiotensin 2, if it is produced overly produced, then that causes lot of problems. Okay, that is one strategy. Another was modulating the calcium channels, the calcium channel blocker, or sometimes some beta blocker. Beta blocker means this beta adrenergic receptors that also can control the flow of blood in the heart, towards the heart and outside. Okay. But apart from this, another factor which is also important is that persons suffering from having a high cholesterol level in their blood. That cholesterol after all has a huge hydrophobic skeleton. Okay. So, its solubility in the biological system, biological solvent system is not very high. Okay. So, if that is not very high, although there are molecules which transmits this cholesterol from one place to another, like the low density lipoprotein LDL, then HDL, these are there which carries the cholesterol from one place to another. So, if there is high concentration of cholesterol, that means the cholesterol limit is crossed over the normal limit, then what happens? You can have cholesterol deposition in the arteries, and if you have cholesterol deposition in the arteries, so your passage becomes narrow, flow of blood will have lot of hindrance, passing through the same amount of blood, but through a narrow passage will result in a back pressure. Okay. So, that is another cause of having high hypertension. Now, remember, even if you lower the hypertension by adding antihypertensive drugs, but if your artery is already having a deposition of cholesterol, that you are not removing, you cannot remove that unless you have a surgery. Otherwise, the cholesterol that has been deposited inside the artery, that will restrict, that act as a vasoconstriction. So, the pressure goes up, and the problem is sometimes this deposition that can come off, like if you take water in a kettle and boil for a long period of time. So, what you see is a layer of calcium carbonate at the bottom. Sometimes what happens, chunks of calcium carbonate can come up in the water when you boil. So, similarly what happens, chunks of cholesterol, mainly cholesterol, this deposit is containing other molecules, but cholesterol is the predominant part. So, if these chunks come off in the bloodstream, now what happens, that can block the valve to the associated with the heart, or it can go and block the nerves, the nerve endings. So, if that happens, then you have in the first case you have what is called stroke, and in the second case what you call cerebral attack. Okay. Heart attack versus cerebral attack. Okay. It's the cause is the same, that it is the cholesterol deposition on your artery. So, this is the picture. Now, I come to the picture. This is the structure of cholesterol. Okay. Now, you see this is the normal artery, there is no deposition, that is the total diameter, inside diameter of the artery through which blood can flow. But if you have higher cholesterol level, continue to have higher cholesterol level in your blood, then some cholesterol will become insoluble, will come out, and then they deposit on the inside wall of the artery. Okay. So, you see as days pass by, what will happen? You have more deposition, and this is an extreme case, that the passage is so narrow. So, you ran the risk of having a heart attack or a cerebral attack. Okay. Now, if you have this type of scenario or this type of scenario where blockage is, say, I will say about 50% blockage or 60%, here the blockage is more than 80%. Okay. This is still okay, but one has to be extremely careful that if the chunks come out, then it can block the valve or the nerve endings. So, the only way you can do, see, one way you can say that I will devise a molecule which will dissolve the cholesterol from the artery, but that is very difficult. That is very difficult, because one which has already forming a crust inside the blood vessel, then that is very difficult to take out. So, what people do is, what is the surgery that it takes place, that it creates a bypass, so that the blood flows through another channel, or sometimes they dilate the blood vessel, that is what is called balloon angioplasty. Angioplasty means if you make it even bigger now, so by putting pressure, and then so what happens, this whatever narrow is there, that becomes bigger. So, it depends on that is of course the decision of the medical doctor, but what we are interested in here, that how to maintain the cholesterol at the normal level. If you keep the cholesterol at the normal level, then your artery will be similar to this. So, the question is how to design basically lowering of cholesterol, that means anti-cholesterolemia or anti-cholesteremic anti-hypercholesterolemic drugs. The meaning is basically nothing, it is a term, but meaning is that how do you, hyper means higher value of cholesterol, how do you lower the higher value and maintain it to the normal level. So, today we are going to discuss this aspect, that how to manage the cholesterol level in the blood. Now, if you want to manage the level of cholesterol, remember we cannot shut down the biosynthesis of cholesterol. You have to attack first, you have to attack the how cholesterol is biosynthesized inside the body. Now, cholesterol can come into our bloodstream in two ways. One is metabolic, that we make our own cholesterol. The other is from the food, that can also come, food of animal origin, that can also contribute to cholesterol, okay, or that can contribute to proteins which carry the cholesterol towards from one place to another. Okay. So, basically what you have to target is how the cholesterol is biosynthesized, is made inside the body, which is, by the way, about 80% of the cholesterol is made inside the body. So, about 20% we take from the food. So, food is not the ultimate culprit, the culprit is the metabolic disorder that is present in the body. Okay. Now, if you then, that means you have to hit this metabolic disorder and somehow maintain, somehow manage it. So, if in order to do that, you have to know how cholesterol is biosynthesized, what are the steps for biosynthesis of cholesterol. If you know that, then you can say there are maybe say 30 enzymes involved in the biosynthesis of cholesterol. Now, by the way, cholesterol is biosynthesized from the same starting unit as the polyketides or the fatty acid, which is acetyl coenzyme A. That is your starting point. Okay. Now, suppose you say that I will stop the biosynthesis of cholesterol, shut down the biosynthesis of cholesterol by having an enzyme inhibitor, which is an irreversible inhibitor. That means, which completely shuts down the enzyme, but that is not what is wanted here, because cholesterol is also an obligatory ingredient for other body functions, like the cell membrane has lot of cholesterol in it. Apart from the lipid, in the lipid bilayer, you also have cholesterol there. Okay. So, that is one, it is a constituent of the cell membrane. Number 2, that cholesterol is the precursor for making different hormones like estrogen, androgen, all these are from where it is being made, from cholesterol itself. So, you cannot completely shut down the level of cholesterol. That means, if your approach has to be to design an inhibitor which is a reversible inhibitor, and the lowering of the efficiency of the cholesterol making enzymes, you have to put at a optimum level. You cannot make it very low, because then you will be starved of this cholesterol which is required for other body functions. Okay. So, that is the case. Now, let us see what are the enzyme systems or what are the reactions that go on in cholesterol biosynthesis. Okay. Now, there is another class of compounds which is present in the in plants, and they are called terpenes. Terpenes are also very similar to the cholesterol, only thing the cholesterol has a phenanthrene ring, a fully dearomatized phenanthrene ring attached to a 5-membered ring, that is called cyclopentenophenanthrene skeleton. On the other hand, or parhydrophenanthrene skeleton, because every double bond is hydrogenated, and terpene is usually 5 6-membered rings fused together. So, that is the difference between terpene and cholesterol. The biosynthesis is very similar, but at some point they branch out. Okay. So, let us see, we are not talking about terpenes, let us see we are talking about cholesterol, but as I said, the branching point up to means this also covers some of the biosynthetic aspects of terpenes also. Later on, if somebody asks you that what is the or is a precursor for terpene biosynthesis, the same thing. Okay. The precursors are same. Okay. Now, let us see what are the reactions. Now, cholesterol structure I have already shown to you. This is the structure of cholesterol. If you count the number of carbons, then you will see there are 30 carbons. Okay. There are 30 carbons. Okay. So, that means 30 carbons, and 30 carbons in case of terpene is what is called triterpene. So, basically it all starts with a C5 unit, which first goes to a C10 unit. This is what is called a monoterpene. Then the C10 goes to C15. So, that means first two C5 units condense with each other to give C10, then the C10 condenses with a C5 unit. We will come back to these units, are they same or different, we will discuss that. You get C15. Okay. That is what is called that is a sesquiterpene. Sesquiterpene is the 15 carbon containing compound, and then 2 C15 combines and you get what C30. Okay. This is either could be triterpene. Based from this C13, the triterpenes could be obtained, C30 means unit whatever, and then that also gives rise to steroids. So, the branching point is here, from C30 you branch out to either to the terpenes or to the steroids. Okay. And then before that the names are actually in terms of not steroid, in terms of terpene, because steroid is only name steroid is given to C30 30 carbon containing compounds. Okay. But there may be some cases where the carbon may be less than 30. I should also warn you that because cholesterol has a side chain here. So, it may be possible that these are totally oxidized by the system and it ultimately becomes a OH or ketone here, then you do not have 30 carbons, but you see the precursor is 30 carbon. So, steroids are either 30 carbon containing compounds or it could be compounds which are derived from 30 carbon containing compounds, where the side chain is oxidized, is broken down, is degraded. Okay. So, you should not go away with a message that all steroids have 30 carbons, that is not true. Okay. Cholesterol has 30, because it is almost the immediate precursor of C30. Okay. Now, let us see what are the reactions that take place. I told you the precursor is acetyl coenzyme A. Now, this chemistry will be easier to understand because we have already gone through the chemistry of the polyketide. Okay. In polyketide chemistry, you know, two acetyl coenzyme, one acetyl coenzyme A and a malonyl coenzyme A, they combine and form the acetoacetyl coenzyme A. But here interestingly, two acetyl coenzyme A combines directly, it is not a decarboxylative pathway. It is not a decarboxylative pathway. What happens that there are bases. See, basically this is an ester, that is an ester. So, ultimately you need a base, so that one goes to the enolate and the enolate then comes and attacks the carbonyl. Okay. I write that. So, you have CH3, then CO, SCOA, thioesters are usually very labile, but the other part it is a hydrogen here CH2 CO SCOA. So, now in the enzyme system you have a histidine which is a base. So, that abstracts this hydrogen. So, this then comes and attacks and that goes first to the oxygen, comes back and SCOA goes out. Okay. However, the actual mechanism is little bit slightly more complicated in the sense that the first attack onto an acetyl CoA takes place by a cysteine sulfur. That means, the enzyme is like this SH. So, that first displaces the thioester, this acetyl CoA is first displaced. So, you get CH3CO and I can write this S enzyme. So, instead of the coenzyme you have a sulfur containing the enzyme, okay. And then the other part which is enolized by an imidazole base, that comes and attacks this compound, means that is the precise mechanism. But in just for simplicity you can say that two molecules of acetyl CoA condenses to form what is called acetoacetyl CoA. Now, in case of polyketide, this gets either reduced or this will be extended, another malonyl CoA which is the extension unit. In this case, another acetyl CoA, there is a third reaction, a second reaction. This first reaction between two acetyl coenzyme A and the second reaction is another acetyl coenzyme A reacts with this carbonyl and makes it into a OH. This is also will be catalyzed by, because you have to abstract this hydrogen. So, you can tell that if I ask what is the possible active site amino acid to pull up the hydrogen, that could be imidazole, that could be arginine, that is also true, but what has been found, it is imidazole. Okay. Imidazole means it comes from a histidine, and then here you have to have a hydrogen donor, because you know that addition to the carbonyl is catalyzed by acid. So, a hydrogen donor will be better and that will be in the form of a BH plus, a protonated base. It could be imidazoleum cation, okay. And that gives the hydrogen to this developing negative charge on the oxygen. So, if that be the reaction, then what you will get? You will get now alongside this you also have a hydrolysis of this acetyl CoA, and if acetyl CoA is hydrolyzed, you will get what? Acetic acid. So, basically this was earlier acetyl CoA. So, CoA leaves and then you get the free acid. Okay. This is what is called beta hydroxy. See, this is a dicarboxylic acid, 1, 2, 3, 4, 5. 5 carbon dicarboxylic acid is known as glutaric acid, and you have a hydroxy here, and that is your, this is alpha carbon and that is beta carbon. So, this is what? Beta hydroxy beta methyl glutaric. This is not glutaric acid, glutaryl CoA, and that is what is called in abbreviation HMG CoA. Beta hydroxy beta methyl glutaryl coenzyme A. Okay. Now, so this is basically in this, first there is a condensation, expulsion of one acetyl, just the CoA is expelled, and then the next step you have a nucleophilic addition to the carbonyl, to the beta carbonyl, and by another acetyl coenzyme A, but that is associated with hydrolysis of the CoA also. CoA also goes. So, that one end you have a CO minus and the other end you have the CoA, that CoA thioester is there. Okay. This is what is called hydroxymethyl CoA, glutaryl CoA, HMG CoA. This is very important, that is why I am spending so much time. Now, what happens? This whatever the thioester was, that is now reduced by a NADPH mediated enzyme. So, you will get a compound where the ester is reduced to the alcohol. That means, it is a reduction from acid to alcohol. So, it is a first goes to the aldehyde and then goes to the alcohol. So, it is a Na, they have found that it is a NADPH mediated hydrolysis, but 2 molecules of NADPH will be required, because you are doing 2 reductions from acid to the aldehyde and aldehyde to the alcohol. Okay. So, this is the compound. This is, by the way, called mevalonic acid in the biological system, because it is present in anion. So, that will be called mevalonate. Okay. I can again, so this is the mechanism. Initially, I told you that when the first reaction takes place, maybe not this one, let us see, I can write the mechanism, that is better. So, the first reaction, the first condensation was like this, a cysteine displaces the coenzyme A, then another CO, SCOA, and there is a base which is the immediate that histidine, sorry, this is hydrogen, and the base is here. So, base abstracts the hydrogen, this goes and attacks, and ultimately the cysteine leaves. So, you get the first compound, acetoacetyl coenzyme A. The second reaction, what happens? That is, the second reaction, you have again a displacement of the acetyl. See, there is another acetyl CoA now has to react with the carbonyl, but this is again already the enzyme has displaced the CoA. Okay. And then again the base, very similar reaction, the imidazole goes there and this abstracts a proton from the base. Okay. So, that will give you what is called hydroxymethyl. See, what is the product here? If you write CH3, a carbon with CH3 OH, and then you have on one side CH2, then CO S enzyme, because still that is attached to enzyme. On the other side you have CH2 COS CoA. So, this is the status. Now, water comes and hydrolyzes this. So, what you get is the HMG CoA. So, HMG CoA is CO2 minus, and then this is Now, interestingly, the configuration, that is also important. It is a stereospecific reaction. You do not get, because you are developing a chiral center here. Okay. So, that is CO2 minus and this is COA. Okay. Now, if you look at the configuration here for the HMG CoA, what is the configuration? What is your number 1? This one is number 1. What is your number 2? Because it ends up with a sulfur ultimately, COS, here COO. So, this will be your 2 and that will be your 3. So, what you get 1, 2, 3, but your fourth group is upwards. So, that will be S HMG CoA. Okay. What is the next step? The next step is reduction of this thioester into the alcohol. Okay. So, that is given here, although the stereochemistry is not given. Stereochemistry is, this is also interesting, that the stereochemistry remains the same, because you are not touching the stereogenic center in this reaction. The NADPH mediated reduction is basically touching the thioester. Now, but if you want to do RS now, you will see this is 1, that is 2 and that is 3, because this is alcohol now. So, now it will be R mevalonate. So, R mevalonate, but that does not mean that HMG CoA has opposite. It has the same configuration, absolute configuration. It is just the priority sequence order that has got changed. So, R mevalonate is formed. Now, what happens? This is how many carbons? If you count there, 1, 2, 3, 4, 5, 6, but still this is not the that C5 unit I was talking about, C5 unit. How the C5 unit is formed? You can form the C5 unit if you do a decarboxylation, decarboxylative elimination, like CO2 minus and then make a good leaving group out of OH. So, phosphorylation of the OH. So, that will, so this is the structure. So, you have the CH3 OH and then CH2 OH and CH2. So, that will be phosphate now. You have to do phosphorylation because OH is a bad leaving group. So, now this goes here, that goes there, that goes out. Okay. But before that, this OH, what has been found? This OH is diphosphorylated. So, the elimination takes place first by putting a phosphate onto this primary alcohol, then another phosphate, diphosphorylation. These are all ATP mediated reaction, and then so it is diphosphate now. Now, this undergoes again phosphorylation. So, you have the leaving group O phosphate and then this decarboxylation I was showing, that takes place. So, what you will get? You will get a compound like this. What is the name of this compound? See, this is a 5 carbon unit, that is one important point. 1, 2, 3, 4, 5. This is a 5 carbon unit. Okay. This is what is called isopentenyl pyrophosphate. Isopentenyl pyrophosphate. There is a delta 3. Delta 3 means position of the double bond. See 1, 2, 3. So, the double bond is at position 3, or you can say isopentenyl. Isopentene means that the double bond is on the terminal end. Okay. So, isopentenyl pyrophosphate, but this is what happens, this can equilibrate between two tautomeric forms. This hydrogen can undergo 1,3 shift and you get the double bond inside. So, that is what is called, this is your alpha, this is your beta, this is gamma. This is what is called gamma-gamma dimethyl, that should be written gamma-gamma dimethyl allyl pyrophosphate. So, basically out of this reaction, that 3 condensation of the involvement of reaction between the 3 acetate units led to the HMG CoA, and then HMG CoA undergoes phosphorylation, bisphosphorylation and decarboxylation, and ultimately what you get is mevalonate, a mevalonic acid which is in the R configuration, and then there is this that through the decarboxylation, what you get is isopentenyl pyrophosphate, which is in equilibrium with gamma-gamma dimethyl pyrophosphate. Okay. So, these are all 5 carbon units, C5 units. Okay. Now, these C5 units, before we go to the next session, I think I will finish up here by saying that these 2 units, this is again the same thing, this is gamma-gamma dimethyl pyrophosphate, allyl pyrophosphate, and this is isopentenyl pyrophosphate. Okay. Now, first of all, you have to identify this part where the functional group is present or the polar moiety is present, that is what is called the head group, and where the non-polar or the lipophilic part is present, that is called the tail. So, this chain isopentenyl pyrophosphate has a head group and has a tail end. Okay. So, for this also the head is here and the tail is here, and the tail carbon is these methyls, and that the head carbons is this allyl, this carbon, allyl carbons are the head carbons. Okay. So, now these two, so head and tail you have to be very careful. In some books it is written in the opposite way, but usually in a fatty acid the head group is the acid group and the fatty chain end group is the tail group. Okay. So, now there will be a condensation between these two, there will be a reaction between these two, and in the next session we will start from here. So, we have discussed that the formation of the 2 C5 units which are in equilibrium with each other. Remember, these are the 2 most important units, one is gamma-gamma dimethyl allyl pyrophosphate, another is isopentenyl pyrophosphate, which comes from the R mevalonate. Okay. Thank you.